What level of the spinal cord does this represent? Pardon? Sacral. And is there a sex difference between the sacral cross-section of the spinal cord of a male and female? What two fiber tracts run in this area that are important to autonomic function? And what rex lamina is this region here and its importance? What level of a cross-section of the spinal cord is this? And what about the posterior column? What level is this? Identify this portion of the spinal cord. Who would like to volunteer? Is this gray matter or white matter? And what column is it? Okay, so gracilis and cuneatus. What column is this? What sensory modality is carried in the posterior column? If we remove this completely at C5, what would be the functional loss to the patient? Loss of position sense. What other modality would be lost? Vibration, position. What else? Graphia. What else? Two-point stimulation. Would those all be gone if we cut that out? What is the emerging concept there? If we took the whole posterior column out, would a person be absolutely deficient in those modalities? Vibration, position, joint position, limb position, two-point stimulation, vibration? No, they won't be. What they will lose across the board is speed of movement. So that introduces that we don't completely understand all the pathways, doesn't it? And that's what, in part, we're going to touch upon. So this, according to Head, was epicritic modalities, in contrast to protopathic, which is pain, temperature, crude touch. Is there anything else that is carried in the posterior column that is not the classic epicritic? We'll go into it. There is one other aspect of pain that is conveyed there. What part of pain is conducted in the posterior column, in the midline, especially on the ventral aspect of the posterior column? Visceral pain. I'm going to go through. At another time, we'll add some more questions. There's a kind of a simple contrast between the cerebellar, and the spinal cord. And it's so obvious, I really didn't think about it too much. There's some good reasons why it presents itself in this manner. But in the cerebrum, the gray covers the white matter. In the spinal cord, the white covers the gray. Does anybody have some thoughts about why that exists in that contrast? Say it louder. Well, probably the simple, without getting into details, is the projections of the long tracts, ascending or descending, are migratory. And the first cells to develop are really more nuclear in the spinal cord. It's more complex than that in the cortex. So from the brainstem down to the spinal cord, because of the development of fiber tracts that occur after the nuclear centers, which predominates from the germinating aspect of the ependema, that may account for part of the difference there. Well, we'll keep it elementary at the beginning. Rostral, caudal, portion of the spinal cord. Cervical and lumbar enlargements, you know why they exist, in contrast to the thoracic, relatively narrow portion of the spinal cord. What is the reason for the cervical and the lumbar? I think you all know that because of the plexuses to the upper and lower extremities, your motor pull is much larger in those areas. It is not that significant in the thoracic area because you have some of the axial muscles and the muscles that make up the truncal abdominal area. Well, let's take a look at some of the aspects of phylogenetic spinal cord variations. If you take lizard, alligator, and mammals, it's characteristic that there is a cervical and lumbar enlargement. They're all tetrapods. When you get to a non-tetrapod, where it stands on the phylogenetic tree, doesn't always agree, so it's a variation depending on the development of the extremities. Well, as you know, the snake does not have the limb. In contrast to some of the fishes, depending on how much they use their fins, there will be the same type of enlargements that are parallel to an appendage, either homologous or imitating. The teleos is interesting because it does something different in its function than spinal cords are expected to do. Who knows what the urophysis is? Urophysis. It's an organ on the caudal end. It's most noted in teleos, and it will produce basically two categories of hormones. One is called urotensin 1, the other is urotensin 2. Urotensin 1 is like corticotrophin releasing factor, and urotensin 2 is like somatostatin. And in some animals, it controls prolactin and TRH. It has recently been noted to be in humans as well. Then it's a bony fish. It's the first bony fish, in contrast to elasmobranchs, which are cartilaginous. Does this really have a clinical importance? I can't really say that it does, other than what we find in regards to their vertebrates and non-vertebrates. I think we all appreciate it's playing a bigger role in how we understand the human molecular connections and the homologies. Interesting, in turtles and birds, the cervical area is very large, lumbar area is relatively small. Why? Because if you look at the legs of a bird, it's not too much, and they don't use their legs a lot other than when they're making a landing. The pigeon is interesting. Pigeon is interesting. It's got a large cervical and a pretty large lumbar, but it has a cavity, and it's a lumbosacral or a rhomboid sinus, and running right through the center part of it is a central canal that connects from one end down to the other and then continues. And that is a glycogen storage. It's believed to be a source of glucose conversion, glycogen to glucose conversion for energy. Now, you've seen this before, and I'm not going to spend much time on it other than on the ventral side of the developing spinal cord, in contrast to the dorsal. Sonic hedgehog is important and in high concentration, and then by diffusion gradients, it is, as expected, will decrease. So from ventral to dorsal, the sonic hedgehog concentration decreases. And BMP, bone morphogenic protein, and there's a series of isoforms, which I won't go through, is increased in the dorsal aspect, especially along the ectoderm, and then decreases in concentration. This sets off, in part, it participates in which one of the morphogenic transcription factors are going to be expressed. And depending on which one of those is expressed, then we start to get a different pool of neurons, especially motor neurons. Jessel at Columbia has done considerable work on this, and that team has shown some really very profound topographical organizational motor neuron pool expressions. For example, in the motor columns are instigated by the relative ratios of transcription factors for the medial motor column. The most medial aspect of it goes to our axial muscles. And that's the motor neuron pool that's dedicated to those muscle groups. And then you can see it's broken down to lateral motor column, a medial and a lateral aspect. The medial aspect goes to the flexor muscle groups. The extensors get it from the lateral portion of the lateral motor column. And there's a special unit here that is the preganglionic fiber that comes from Torney's neuronal preganglionic pool. All these are dedicated by the preceding ratios of transcription factors. This is an example. This is taken out of Carpenter and Truex in an old edition. I haven't been able to find it in the last edition, but I think it's very interesting, of the central canal in the cervical area of the embryo. You can see the length of the ependymal cells. This is a newborn from the sacral area, and this is cervical of an adult. And you can see that there are changes there. I wouldn't say it's a study, but it's I thought interesting demonstration. Now, the perspective I think that many of us have when we think about the spinal cord is a cross-section. But more than that exists, and it doesn't really, this topographical representation of the spinal cord isn't true to what we really should see when we operate. And the simple story is this. Here's the median central sulcus, and this is the white matter, and this is the gray matter. Neuronal pools and the posterior horn do not exist as segmental nuclei. Now, I suspect everybody appreciates that, and that's not an epiphany for anybody. But I think it's hard for us to hold that in our mind, that the motor pool and the rex laminations in the dorsal horns are one continuous series of nuclear elements. They're not segmental. Now, in some animals, they are segmental, but not in humans. This is to show the medial and lateral dorsal root divisions. That if one went from the periphery to the base of the posterior horn, this is a very important area for the medial and lateral dorsal root divisions to be appreciated. The medial division has predominance of two types of fibers from the periphery, first order neurons. There's the A alpha and the A beta. These are rapid conducting neurons, as you know, and they're in the vicinity of 50 to 120, maybe 150 meters per second. They're heavily myelinated, and as you see, the dorsal root ganglion, their perikaryon is large and pale, and they pass just dorsal to the tract of Lishauer. The tract of Lishauer is an ascending and descending 1 to 3 vertebral segments of ascending and descending before it enters the gray matter to synapse. What disease is classically known and is associated with extreme pain? Syphilis, Tabes dorsalis. That's one of the early areas of Tabes dorsalis, and it probably more than clinical has some experiment by biology, by nature, importance to it. Now, the lateral division are A delta and C fibers. These are like 10 meters, and this is like less than 1 meter per second, and they enter directly into the gray matter. Rex laminations. Are you familiar with Rex laminations? Does it mean anything to you? Well, around 1950, 1952, Rex, a Scandinavian neurophysiologist, neuroanatomist, further classified each level by its cytoarchitecture. So this was based on a Nissl stain. It was very tedious work. It was better than a decade of dedication. And one way or another, this classification has remained. And there are some deficiencies to it, and there's some argument about is layer 4 to one person, layer 5, lamination 5 to another. And it's not that important because it does carry some functional significance, especially in nociceptive pain studies. But this is only for the nuclear elements, and it's the characteristic, the cytoarchitecture of the neurons. The tract of Lishauer is out here, and that's a dorsolateral. It's defined as the dorsolateral segment. Then there's lamina 1, very important for pain. And then lamina 2 is really the substantia gelatinosus. The best way to put it together is lamina 3, 4, 5, and 6 together are really the proprius. They're the principal sensory nucleus of the posterior horn. This is where the modulation, the first order and second order neurons are having their synapses, and the modulation that occurs either intraspinal or by supraspinal input. And probably to group 3, 4, 5, and 6 together is not a bad idea from the standpoint of function. And then 7 is a little bit more complex, but the best way to remember 7 is the intermedial lateral portion in the thoracic area is where you find your autonomic, your sympathetic preganglionic fiber. And the dorsal aspect is Clark's nucleus for the spinal cerebellars. And 10, to remember, has multiple synapses, but probably, this should really be 10, that's an error there, is for what I will discuss briefly with you today is the propriospinal system, which is an important system that is coming into its own study. The lamina 9 is really the motor pool. And you all know about the motor pool. If you want to have a generalization, if we stood here and said that this is just the mature state of the motor columns, therefore those that are most medial are going to be in the thoracic area, predominantly axial muscles. In the areas of extremities, then for upper and lower extremities, it's going to be proximal and the paraspinous muscles. That's the motor pool for that. And as we go out more distally, then we get extensors and flexors. And you've all seen that picture in the textbooks of the person flexing the arms. Just remember this, that's a composite picture of the motor column. You never get that representation of hands, forearm, shoulder, biceps in one section. So it's part of the motor column that's built together to give you that topography. The X's around here are fascicles of the propriospinal tract. The propriospinal tract only exists in the spinal cord. It's an intraspinal projection. It never goes into the brainstem and above. And the fascicle septal marginalis and the fasciculus interfascicularis, they may be carrying those fibers. And they're second order fibers. They're not part of the first order fibers of the medial lemniscal, posterior column, medial lemniscal system. The two sensory systems that one can generalize about is the posterior funiculus hyphen medial lemniscal system, which is by Head's description is predominantly epicritic. And then the crude pain, temperature, itching goes to the anterior lateral system, the spinal thalamic systems, which I'll get to in a second. But the parafascicular area and the propriospinal tracts that are short and long, and I'll show that at the end, and these interfascicular and septal marginal have not been clearly physiologically stated, but they're probably second orders. They go up several levels and then synapse to go into this area here, the second order, and those are visceral, for visceral pain perception. This is just to show there's a population of larger neurons, and the largest ones are just what you would expect. And the motor pool that goes to the extremities. This is the thoracic area. So you see the intermedial medial, and you see the intermedial lateral portion in 7 in the thoracic area. And we, I think, all know and appreciate that the lateral portion of the intermedial lamina 7 is preganglionic. And that's an interesting relationship, isn't it? It's an anterior horn cell equivalent in the intermediate horn that is not neurocrest that goes to neurocrest, because the postganglionic or the ganglionic neuron is of neurocrest origin. Does anybody want to take a stab at some of the suspected functions of the intermedial medial portion of 7? Respiratory. It's sensitive to respiratory. Now, here's a general circuitry for fast pain and crude touch in the dorsal horn, and the importance, forgive the spelling there, of the WDR, which is wide dynamic range neurons. I'm going to somewhat be careless here. I like to think in pain conduction of the spinal thalamic anterior lateral system. Remember what I said. There's a posterior system that is for epicritic in the classic description, for vibration, position, 2-point stimulation, graphia, speed of movement on the cutaneous area. And then we have the anterior lateral system, which is spinal thalamic, with some variations on that theme, which I'll go into. And then we have that diffuse reticular system. I like to think of the DWR, which is in the lamina 5, as kind of like the final common pathway issue to the conduction of spinal thalamic. And then we have that array of discussion of an array of physiological points that people argue about the modulation within the spinal cord, which will continue at the end of the talk as to gating theory. So we have the alpha beta, which is a fast conducting around the 50 meters per second, and the A delta, which is 5 to 10 plus or minus meters per second. And this delta comes in on the lateral division of the posterior root, dorsal root. And then we have an A beta. A beta is fast, and it has to do with cutaneous input. It can be Pacinian corpuscles between the lower end of the dermis, and that tells us when our skin is moving, or a Meissner's corpuscle, or a Ruffini's, or Merkel's, and these things will tell one about if the skin is being touched. It's very accurate, and that these two come in, and they enter lamina 1. Lamina 2 is substantia gelatinosus. And in lamina 2, it really does not project any place. Lamina 2 does not project to supra or infrasegmental levels to any significant degree. It does not go to the brainstem or to the diencephalon, telencephalon regions. But what it does have is a series of inhibitory cells. And those inhibitory cells are the endorphin mediated cells for their terminals. The descending tracts, like a type C, could come down to this in lamina 1 and contribute to the spinal thalamic, and crude touch from the A beta synapse, and those are in 4 and 5. But what the WDR does is it has a heavy ramification of its dendrites into the upper levels. So if A delta comes in, which is a pain conductor, a nociceptive conduction, it will activate an inhibitor of an inhibitor, which is what? Disinhibition. So it's excitation. And so that ends up exciting WDR. And why do they call it wide dynamic range? It's because it gets information not only from pain, but it also gets information from an A beta, which is going to do what? It's important because if that's theoretically and in fact not present, the subject detects pain, but they can't localize it. So this is an important adjustment for localization from the A beta. And the A delta as inhibitory to the inhibition to excite the WDR, and at the same time the A beta, which is for position to know where the instigating noxious stimulation came from, because this is an innocuous. It's innocuous in contrast to noxious that comes from A delta. It is inhibitory directly to an inhibitory in lamina to keep the WDR neuron quiet. So that's an important fact, and here's a kind of a summary. And I think this is general knowledge, but it's something to hang your hat on for kind of the pragmatism of residency, and that is for your boards. If you talk about a motor nerve, generally the classification is 1, 2, 3, and 4. 1, 2, and 3 are myelinated and decreasing magnitude, and down here for unmyelinated. A good example of this would be like a postganglionic. And you can see that the dimensions and diameter are decreasing. And the rule is, with decreased myelination, you get decreased velocity of conduction, or said a different way, the more the myelination, the thicker the nerve diameter increases the conduction. Now, if you take a nerve and you stimulate it, you can separate out A alpha and A beta, which are fast-moving, innocuous, non-nociceptive fibers. They're the fibers for your stretch reflexes, for a non-painful stimuli that come to the skin. And you get an A delta blurb here, and depending on where you are in the nerve or where you are in the spinal cord, these can separate out. But you can see between stimulus and 10 milliseconds, this is the group of spikes that you get. This one would be involved with pain. C is involved with the burning portion of pain, and A delta is the sharp aspect of pain. And you see that these are very, very close together. If you burn yourself, accidentally touch a hot oven, that first pain is very sharp. And that is being conducted by the A alpha and A beta, especially the A beta and to some degree the A delta. In time, what happens? That becomes a very annoying burning pain. So there's a sharp pain, and then there is the burning pain. And the electrophysiology of those also are correlated to the fibers and the peripheral receptors. Now, if we take the thalamus and we break it down a little bit more, the thalamus receives two parallel nociceptive projections from the anterior lateral system, the spinal thalamic tract as you know it. And we don't differentiate so much between, anterior and lateral. There was a time that was popular. That's no longer focused upon. One is referred to as the medial group, the paliospinal thalamic projection, and the other is the lateral group, the neospinal projection. This is in part evolutionary as well. If we take for localization in the primate, which is late in the mammalian evolution, you have the neospinal thalamic system, and it's focused on the wide dynamic range neuron, secondary neuron, activated by primary neuron A delta in lamina 4. It goes to the ventral posterior lateral and posterior thalamic nuclei. It then settles into anterior cingulate and insular cortex, and we'll go a little bit more into some other projections. So this is the new system. The older system is the paliospinal, and it's involved more with the emotions. So emotions to pain preceded localization. And it is predominantly the C fiber primary neurons activating secondary neurons in lamina 7 and 8. These have projections that are predominantly to the interlaminar central thalamic nuclei, or parafascicular nuclear group, or centromedian region, and some of the reticular formations surrounding the thalamus. They project to the anterior cingulate and insular cortex. Now, I always like to make things complicated, and this is taking the slide before and just making it a little bit more complicated, but redundancy is a good foundation for retention. If we look at the lateral portion of the anterior lateral nociceptive system, the neospinal thalamic that developed in primates, that is predominantly WDR. It goes to the posterior nucleus. The VPL projects to primary somatosensory, a lot to supplementary, and it's sensory discriminatory. If you take the medial division, is paleo spinal thalamic, some like to say spinal reticular thalamic, developing in early mammals. It's a slower system because it has many synapses through the reticular formation as it goes up. And it's midline thalamic nuclei. It's C-type of primary first-order neuron from lamina 7 and 8. And it goes to insular cortex, anterior cingulate, and it's affective and motivational. This is much more in that complex area of pain that we see clinically. And I didn't put all of the other areas that it goes through, the amygdala, the thalamus. Some like to say that they're in the ascending pain pathways. There's spinal thalamic, there's spinal reticular, there's spinal mesencephalic, there's spinal brachial mesencephalic, and spinal hypothalamic. And the short story, and it's accurate, when you put spinal thalamic, paleothalamic, and a third category of multiple ways of getting to the brainstem and higher, even to the cortex, that it is such a diverse, such a complicated pathway that has evolutionary justification that we have trouble dealing with it clinically, because it is a short benefit to do tracheotomies, nuclear injury, et cetera, because there are so many different pathways, and some of them are even more complicated than where the pain is, how sharp it is, and if you rub on it, it goes away. It's very hard to rub on the amygdala, insular cortex, anterior cingulate with all the Pavlovian, et cetera, inputs that we have in our life, which makes the perception of pain much different. And I think Langston and Dr. Hawley could give a lot of examples of how that becomes almost impossible to figure out. Now, this is when you put animal studies together in midline myelotomies, in the thoracic area, I chose the thoracic area because it's the best known, dorsal root ganglion, coming in with its medial division. This area is for visceral nociception. No, it's spelled correctly. This area receives from lamina 6, 7, 8, maybe 10 second order neurons. So we have second order, not first order neurons in our posterior column, which traditionally has been what? You go to the lateral division of the posterior root, going into the posterior funiculus, and then going to the medial lemniscal system, cuneatus gracilis, crossing over at that point through the internal arcuate, et cetera, to the thalamus. So it's contralateral. Here we don't have that. What we have is a second order group of neurons, and it is involved with visceral pain. And if we looked at the dorsal root ganglion, it has a little different character for these visceral pain receptors that are first order. They're not small, they're not large, they're intermediate. And they, for the most part, have the same staining characteristics as other pain fibers. So It's basically size that has been studied. And this is for the midline myelotomy. Now, C fibers signal heat, pain, and itching. The only thing I will say about itching is that pain fibers may actually inhibit a pain production from itching. They say that it is rare to have itching and extreme pain at the same time. And some have argued, and there are some animal studies that support it, that it's carried by C fibers. C fibers will take and activate an internuncial to inhibit a pain fiber. Now, I don't know how true that is, but it's an argument against pain physiologists. But the response of warm, non-nociceptive fibers to increasing temperature decreases as the magnitude approaches induction of pain response. In contrast, the C fibers responses increase. This is paralleled by the human subject's subjective response. What does this say? That if you take a non-nociceptive heat-detecting fiber, that at 40 degrees, it's giving the maximum amount of frequency of discharge. And then if you carry it further, it drops off. That is different than a C nociceptive fiber, which increases its firing and is paralleled by the subject's reporting of discomfort. Now, C fibers are different. I'll ask you a general question. It's a conceptual question. Do all sensory first-order fibers report pain? Answer, no. C fibers and A delta are the ones that carry pain, where C fibers are a bit different. They're polymodal. They will respond to heat, they'll respond to acid, they'll respond to cold. So in their primitive way, they are a bit more complex. And response in spinal cord in subject's pain. With the bottom line on the experiments on this is a type 2A, A delta, gives when there's sharp pain, it's being conducted along that route. The C fibers are the burning pain. Now, primary and secondary hyperalgesia. There are two forms of hyperalgesia. There's hyperpathia, hyperalgesia, but I don't like the term hyperesthesia. I don't think we ever feel with greater exquisite perception of touch, two-point stimulation, etc. But what can be is when we talk about pain, if you take the primary zone of injury, which is responsive both to mechanical and heat stimuli, and you take the penumbrum around it, which is a secondary zone, which is the uninjured tissue. So the red is the injured tissue. The green is the surrounding uninjured tissue that will demonstrate hyperalgesia to mechanical but not heat stimuli. The center portion, the point of injury, will respond to both. And that's what these curves indicate. Now, primary hyperalgesia is said to be from tissue injury that's basically non-neuronal. In contrast to secondary hyperalgesia, which most of the time has neuronal injury. I won't argue with that, and I think it's a point of reference that you should keep. To me, all pain is neuropathic, because we can't sometimes figure out what damage that a non-neuronal injury has caused the neuron. And in a way, this will display it. Let's take the so-called primary hyperalgesia axon reflex, which is, in my way of thinking, an efferent action in an afferent neuron. What do I mean by that? That a sensory nerve, a C neuron that goes to the skin, is afferent. It will follow the Bell-Majundi law, which you all know. Posterior root is sensory, ventral root is motor. Not quite. Ventral root does have sensories, more than a loop of sensory. Some horseradish peroxidase experiments show that the ventral root really does have some sensory fibers in it. Well, here's a posterior root, which Bell-Majundi's law would support. It's sensory, but this is a sensory nerve that has an effector function. In what way? The skin gets stabbed. And the next thing is that you got blood and you got platelets. You got release of platelet activating factor, bradykinin, prostaglandins, phosphoinositols, hydrogen and potassium leak out of the cell. Doesn't get enough blood supply, little changes. And that activates receptors on the terminal end of a C fiber, and an axon reflex occurs, which is the simplest reflex that we know, and that creates a release of substance P, a neurokinin, TNF, IL-2, 4, and 6, and CGRP, calcitonin gene regulating protein, that this cocktail of especially substance P and CGRP take the capillaries and they leak. So that's the swelling that occurs in primary hyperalgesia. The mast cells, they respond to substance P and CGRP by releasing histamine, and that is supposed to be a theoretical and in part verifiable mechanism of primary hyperalgesia that continues after an injury that really does not have, from the point of the theory, injury to the nerve. Then we get the central sensitization, which is secondary hyperalgesia. And I think a good example of this is the central cord syndrome. Patient bruises their spinal cord. Next day, you examine the patient, and you take a non-noxious stimulus, as gentle as touching their arm, and you've all seen this that have been on the clinical service to see these kind of patients, and they don't want you to touch it. They don't want the wind to blow. So an innocuous stimuli precipitates a noxious response. This is an accumulation of a lot of different data. It's inaccurate, but it's what's in vogue to discuss. So it's kind of one of the, what that popular term, one of the emerging discussions as to the mechanism. So in lamina 1, if we take an injury to a nerve, and which is predominantly NMDA receptors, glutamate, and when it's injured, when those nerves are injured, they leak ATP. ATP is to be maintained in the cell. When a cell is injured, it leaks out. So it becomes, in a sense, a transmitter of unwanted events. And one of those is activating microglia or astrocytes in the spinal cord, which upregulates purogenic purine receptors, and that stimulates cytokine release. All of these have been named: IL-1, IL-2, IL-4, IL-6, even IL-10 and TNF. And IL-10 is actually an inhibitor of other cytokines. But at the end of the day, these are cytokines and chemokines in some spectrum that are released, and they block the ionic pump, which causes a backup of chloride into the second-order neuron. Now, that increases intracellular chloride. The next thing that happens is that there is always some activity of GABA and glycine receptors that have a certain tonicity. And that may be with unpredicted modulation as well. Now, as you know, if you have GABA and glycine, they generally open the gates for chloride. In this case, instead of chloride running inside the cell to hyperpolarize it, since the chloride has been increased intracellularly, it runs out, and that causes a depolarization to lower the threshold for any leaking glutamate, and that activates the axon for the spinal thalamic. Again, this is all within the realm of discussion, and much of it has evidence, and much of it is not proven. But if you take the pain inflammation receptors and channels to peripheral primary nociceptives, here are some of the representatives at the terminal end of a first-order nociceptive fiber. It's going to have sodium channels that are expressed, nerve growth factors, and a receptor TRK. And then there's TRP. Who knows what TRP is? Transient reversible potential. It's the substance P and neurokine receptor, and it's what responds to heat. It's called a vilanoid receptor if you take to invertebrates. So these are expressed at the ends, and then there's acid sensing ion channel. There is ATP, prostaglandin, and a bradykinin and its associated receptor. Bradykinin is BK, bradykinin receptor is B2. So what is this demonstrating? That the terminal ends of these sensory fibers out in the skin and the muscles, etc., are with a full array for their polymodal response to acid, to potassium, to changes in sodium, to heat, temperature. And here is a similar between the presynaptic and postsynaptic, and I won't belabor it, but what is important is that CGRP and substance P and TRK-B are very important in pain modulation, and I'm not going to belabor that. Here is something that is a bit interesting. If you take preganglionic sympathetic and postganglionic, so-called ganglion neuron, and they go out into the periphery, when there's injury, especially crush injuries, and we talk about causalgia, complex chronic regional reflex sympathetic dystrophy category, reflex sympathetic dystrophy 1 and 2, that something interesting happens. That there's an upregulation of adrenergic receptors at the terminal ends of sensory nerves. This is periphery. This is towards the spinal cord. And in addition, there's an upregulation of adrenergic receptors on the cell body, the perikaryon. And as you know, one of the treatments is that some people do respond to beta blockers. It's not so successful that it's one-to-one, but there are people that will respond to it. And what this is saying is that circulating, if not aberrant, regenerated postganglionic fibers cannot come up and emit their catecholamines around a nucleus that now has increased receptors. Now, Wall and Melzack, they won the Nobel Prize, I think it was between 1964 and 1965. And this is a frequent diagram that they show. And they have touch and pain coming in, and they have a inhibitory to this neuron that's spinothalamic. And since that time, since the late 50s and mid-60s, we know this is the wide dynamic range neuron. And we know that there's more than one inhibitory cell that can inhibit an inhibitory cell. So we have 2 slides and then we finish up for part 1. Possible circuitry for the gating theory. You have low threshold A beta, and you have low threshold, high threshold, this should really be high threshold C-type fibers, and high threshold A delta. When the A beta, which is a non-noxious input, it comes in and immediately activates in lamina 1 a inhibitory to the WGR, which is, in my way of thinking, the final common pathway, second order neuron to the brain, lamina 5, spinal thalamic. So that's a direct inhibitory. Coming off of A delta, in C-type, nociceptive input goes to an inhibitory, which is an example of disinhibition to shut down the inhibition of the DWR neuron, and at the same time, those 2 pain fibers are stimulating. So here's a pragmatic argument from almost a lay person. When it hurts, I rub it. When you're rubbing it, you're activating the A beta. And so the gating theory concludes that in some way that is profitable to inhibit the spinal thalamic second order neuron. But it's in balance and may be overtaken by the A deltas and C type. What can make the difference there? Remember I said there's spinal thalamic, that's neospinal thalamic system. There's paleothalamic, which is really in the order of spinal reticular thalamic, which is polysynaptic, so it's slower, it's more diversified. And then we get that other great diversification that goes to what we call the parabranchial nucleus, which sits in the midbrain lateral aspect and projects to the amygdala and projects to the periaqueductal gray. So there are reinforcing historical events in a person's life that can play a role. You got insular cortex, you got amygdala, you got prefrontal, you got second supplementary cortex, S2. And so if the dorsal root ganglion comes in, and this is a pain fiber, and it's mostly ionic and metabolically inducing, like a G protein, pain information to activate those systems. And that goes to lamina 1, which is one of the origins of the spinal thalamic neuron, 1 in 5. And that is the matrix, lamina 1 and lamina 5, to have pain, and you have an intraspinal gating system. And on top of that, you have the sophistication that goes up to supraspinal levels and descending modulators of pain from the locus ceruleus, medullary reticular formation, nucleus raphe magnus, PAG, periaqueductal gray, hypothalamic spinal, and indirect amygdaloid influence. What this does is it's going to have 5-HT for the midline raphe. It's going to have catecholamine, norepinephrine, from locus ceruleus, but they come down to lamina 2 and activate what? They activate an inhibitory. And that inhibitory neuron is what type of neuron? Correct. It's an opiate. It's an enkephalin releasing, which increases the spinothalamic lamina 1 second order neuron to do what? It increases potassium ionic conductance, so that hyperpolarizes. Now I'm going to end with this. Short and long propriospinal tract. It's intraspinal, as I commented on earlier. It runs on the periphery of the gray matter of the column, the gray column. It has long and it has short. Now, neurologists know about a propriospinal myoclonic disease. Have any of you heard of that? They feel that when this system is injured, especially the long, that it creates a myoclonus that predominates the axial musculature and the abdominal thoracic musculature. And so it's not so much the arms are involved, but jerking of the abdomen. That's a motor system. But it also plays with the infra, supra segmental modulation of pain. And I could not conclude if it facilitates or it inhibits it, so I will just generalize about it. I'm going to stop here. The next part will be on more the motor system and the rex lamination. I fully appreciate that this is not a dynamic topic to some people in the audience. I've tried to water down what I would call an immense amount of data that I can't figure out. And I'm not sure I understand many of their experiments and how they made these conclusions. I have reviewed for some of the journals where people have talked about the visceral origin in the posterior column to pain, and when I read their studies, I can't accept them. I've read other studies that are very convincing, but I think, and probably who has a real significant experience is Dr. Badstorff, doing many different types of spinal cord pain modulating procedures, and I suspect you log some with midline myelotomies. With a Brownstead card, if we make it for points of discussion a very clean cut, an idolized clinical injury to discuss physiology, when nociceptive fibers enter the spinal cord, they enter through the lateral. Very few synapses at that level, and I think this is your point. Some will go up and some will go down. So, if there is a wound to the spinal cord, a hemisection on the right side, the ascending fibers on the contralateral side are going up, and they're spared. But on the side of injury, you're catching and cutting a portion of the spinal cord that's lower. So you're going to have a lower sensory level. But at the level of the injury, if you do a careful examination, the probability will be that you have a band above the injury to the spinal thalamics. So the spinal thalamics on the contralateral side of the injury is going to have a lower level than the point of injury. Should I say that again? Do you understand what I mean by that? So the point of injury is going to be associated with a lower sensory level contralaterally because of the ascent in Glishauer. But at the level of injury, there will be a band because you get the ipsilateral and the contralateral before they have separated. So there will be a band for pain and temperature and crude touch. On the ipsilateral side, you will have posterior column. The probability of a visceral dysesthesia, I have no comments. There's discussion about that, about how the somatic innervation of the abdominal and the thoracic cavity, in contrast to the visceral structures, how they perform with a hemisection. And one of the problems is that they get some sensory from sympathetic and parasympathetics, which throw a monkey wrench in trying to figure out level by looking at viscera. And I will talk about corticospinal and rugospinal next week, to what you will find. So posterior column is ipsilateral at the level of injury. The only time that changes is what we talked about, dissociative sensory findings in syringomyelia, because that's a very selective injury to the second order spinal thalamics, isn't it? Because that's a crossover, usually cervical, which will give the Shaw, quote unquote, distribution, because you're catching only pain at that theoretical isolated injury from a syrinx. It's not catching posterior column or the ascending spinal thalamics. It's just getting the second order neurons that are crossing at that level, so it will just injure that segment, that representative segment topographically. Okay, any other questions? Thank you.